We know that human anatomy constrains brain activity. The physical structure of the brain dictates how signals travel and where they process. Yet, the exact mechanism governing these patterns at a macroscopic scale remains an open question. Modern neuroscience relies heavily on the connectome. This approach models the brain as a complex, discrete network of specialized regions acting as isolated nodes wired together by physical axonal fibers. There is an alternative perspective. The cerebral cortex is a folded, continuous physical surface. It occupies three-dimensional space, possesses intrinsic curvature, and has a specific geometric shape. A recent framework proposes that this physical geometry might be the primary constraint on macroscale activity patterns. In this view, the actual shape of the brain dictates how large-scale signals behave. This framework treats structural geometry as a powerful lens that works alongside our understanding of neural connectivity. It offers a new way to map how signals organize across the cortical surface. Determining whether macroscale dynamics are driven primarily by a complex array of wired connections or by the physical resonance of the cortex's shape affects how we model human cognition. To test this, researchers look at macroscale brain activity measured with functional MRI, or fMRI. This imaging technique detects changes in blood flow to track large-scale regional activations across the entire brain. Because fMRI measures blood flow rather than direct electrical impulses, the resulting data is indirect and noisy. Pinpointing exact causal mechanisms from this information is notoriously difficult. Historically, researchers mapped this data onto structural connectivity networks estimated from diffusion MRI. Diffusion MRI tracks the movement of water molecules to infer the pathways of nerve fibers. However, translating this diffusion data into a complete structural map requires complex, subjective modeling choices. When you map billions of potential interactions using discrete nodes and edges, the resulting connectome models become incredibly parameter-heavy. They require immense computational power and rely on numerous assumptions. This parameter explosion drives the need for simpler models. If a model requires fewer assumptions but accurately maps the exact same brain activity, it offers a more robust tool for analysis. Neural field theory provides the mathematical foundation for this approach. It is an established framework that treats the large-scale cortex as a continuous physical medium rather than a collection of distinct, discrete territories. Highly detailed network models are still required for studying fine-scale, localized brain functions. Simpler geometric models offer a vital mechanism specifically for understanding broad, macro-scale states. The complexity of network models forces us to ask a precise question. Can the physical rules governing continuous surfaces accurately predict the entire brain's macroscale activity? Instead of viewing the cortex as a map of isolated functional zones, visualize it as a single, uninterrupted sheet of tissue folded in on itself. By treating the brain this way, we can calculate its geometric eigenmodes. An eigenmode is a fundamental spatial pattern, mathematically determined by the physical boundaries and shape of an object. Think of a vibrating violin string. Its boundaries dictate where standing waves naturally form. Using a high-resolution mesh, researchers derive these exact same resonant modes from the human cortex. Low-order modes feature broad wavelengths, while high-order modes break into tightly packed granular patterns. The objective of the study is clear. Determine if pure, shape-derived geometric modes can accurately reconstruct the actual macroscale fMRI activity recorded in human subjects. Testing this hypothesis does not claim geometry dictates all brain function. It tests whether geometry provides the principal structural constraint on how signals organize at a macro level. If pure mathematical shape modes can successfully reconstruct biological brain data, the physical anatomy of the cortex inherently guides macroscale functional patterns. To test this, researchers analyzed human connectome project data from 255 healthy participants, including resting state recordings and 47 distinct task contrasts ranging from motor to cognitive functions. To ensure the results generalized across completely different experimental contexts, they tested the model against over 10,000 additional independent activation maps from the NeuroVault repository. The researchers pitted the geometric eigenmodes against several alternatives: connectome-derived eigenmodes, principal component analysis, and an exponential distance rule model, a baseline assuming connection probability simply drops off exponentially the further apart two brain areas are. Across all datasets, the geometric eigenmodes successfully and parsimoniously reconstructed the large-scale fMRI activity patterns. Because geometric modes derived solely from cortical shape capture the data so cleanly, we can achieve highly accurate descriptions of whole brain activity without relying on complex, parameter-heavy network maps. Reconstructing the data with geometric modes reveals a counterintuitive truth about how spatial scale operates in the human brain. Traditional fMRI analyses often apply strict statistical thresholds. This visualizes brain activity as focal, spatially isolated blobs lighting up in specific regions during a task. The empirical data tells a different story. Across 10,000 maps, macro-scale activity is dominated by broad, continuous patterns. The dominance of these long, sweeping wavelengths supports describing the brain using a physical wave model, treating signals as propagating across a continuous medium rather than jumping between discrete nodes. This graphic compares a biophysically motivated damped wave model against a standard neural mass model. When simulating a visual stimulus, the wave model accurately reproduced the brain's temporal hierarchy, tracking the signal propagating outward over 42 milliseconds. Finding that brain activity behaves as a continuous, system-wide wave challenges the assumption that complex tasks are processed exclusively in isolated, localized cortical clusters. The highly folded neocortex clearly demonstrates this coupling between shape and function, but the brain contains deeper, evolutionarily older structures. Researchers extended their 3D eigenmode analysis to the thalamus, striatum, and hippocampus to test if physical geometry constrains activity universally. This chart shows the primary functional fMRI gradients in these regions match their geometric eigenmodes almost perfectly. Looking across the first 20 modes, the alignment holds, even though the functional fMRI data was processed completely independently of the anatomical shape data. The tight alignment inside the subcortex confirms physical shape operates as a universal, brain-wide constraint on function, deeply embedded at every level of our neuroanatomy. The paper establishes clear scientific boundaries. It challenges the idea that connectivity alone explains dynamics, but does not suggest specific neuronal connections are irrelevant. Macroscale geometry is one vital constraint among several. Physical shape guides the broad spatial patterns, but it does not fully determine fine-scale local cognition or individual behavior. For research laboratories, this approach offers a massive methodological advantage. Geometric modes require only a simple structural MRI mesh. This bypasses the need for complex, subjective diffusion tractography pipelines. This computational simplicity opens robust new avenues for analysis. Researchers can track neurodevelopmental shifts, identify clinical disease disruptions, and compare different species. By stripping away parameter complexity, scientists now have a unified, reliable mathematical basis to compare macroscale brain dynamics across any lifespan or organism. If we step beyond the strict claims of the published paper, this physically grounded framework invites several broader theoretical interpretations. First, it impacts the future of brain simulation. Because geometric eigenmodes simplify whole brain modeling, rendering massive, large-scale simulations becomes computationally viable. With these simplified simulations, researchers can test exactly how slight physical deformations in cortical structure, whether from trauma or genetics, alter systemic functional outcomes. This also influences our understanding of cognitive evolution. Physical alterations to the geometry of the brain over millions of years may be a primary, physical driver of changing cognitive capabilities. There are immediate implications for artificial intelligence. True synthetic intelligence may require engineering the specific physical or geometric architecture of a processing space, rather than simply increasing network parameter count and connection density. If biological intelligence is inherently tied to physical resonance, the next leap in computing could involve designing systems where the physical geometry itself is engineered to support complex, wave-like dynamics. The data shows that physical geometry provides an elegantly compact, mathematically robust way to describe the macro-scale activity of the human brain. The detailed wiring of the connectome remains deeply important, but to fully map human thought, the brain must be studied as both a complex network and a physically resonant object. If our brain's physical shape dictates our thoughts, we must ask, what does this mean for the architecture of future synthetic minds?